Well, hello everyone. I'm Dr. David Ajibade, and welcome to Your Health in Your Hands. Now, at the Brain and Body Foundation, we do all kinds of stuff with brain disorders, with hormonal issues. We work a lot with kids with genetic disorders and so on and so forth. But the idea and the topic of microbes, bacteria, and all the others is beginning to gain greater and greater prominence. In fact, when we were in medical school, almost 25 years ago, when I was, bugs, bacteria were thought only as bad. So you have the E. coli, the MRSA, Staph aureus. These are all really, really bad and even ugly infections that we're supposed to wipe out at all costs. So that's why we had to use antibiotics and so on and so forth. But now, in recent years, with increasing technology and increasing diagnosis, we are beginning to find out that there are good bacteria that actually work for us. And as a matter of fact, even beyond that, are critical to our health and development, even from the womb. And this is amazing because it's a total change in our whole paradigm, I believe. And it's going to really become more and more prominent as the years and decades go by in the way we treat patients, whether neurological patients or what you call it now, those chronic diseases, even cancer. So it's becoming more and more prominent. And we've been using what are known as probiotics, good bacteria, to help address disorders such as autism and Down syndrome, genetic disorders, yes, even sickle cell disease, and even in adults, we've seen remarkable changes and improvements, even in things like strokes and dementia and memory loss. Now, these are not treatments, obviously. These are just things to help in the recovery of these patients. So we felt that it would be fantastic if we had someone who really knows about this come and speak to you all about the whole idea of microbes and their place in human health and sickness and disease. And we have none other than someone I've come to respect a great deal, Professor Sarkis Mazmanian. Hi, Sarkis.
Hello, David. Thank you for having me, and thank you for the opportunity to discuss this very, very important topic that you just introduced. I think you did a very good job in framing the change and the shift over the past, I'd say, 10 or 15 years that, at least in the research and scientific community and partially in the medical community, has happened in terms of how we view bacteria and how we view organisms as, again, not these insidious little creatures that only want to make us sick, but ones that potentially can, be developed to improve human health.
Absolutely. And you've been working in this field for quite a while. Can you just give us a brief background of who you are, what you do, and how you came into these discoveries that we'll talk about soon?
Sure. I don't like to talk about myself too much, but my formal training is in microbial pathogenesis. In fact, you mentioned MRSA, which is methicillin-resistant Staph aureus. My entire thesis, my PhD, which was done at the University of California, Los Angeles at UCLA, was studying Staph aureus and its virulence factors, how this organism causes illness. And this was at the end of the late 1990s, when, again, at the time, any self-respecting microbiologist was either studying infectious disease or microbial physiology and metabolism, but there really was no active research in trying to understand beneficial organisms. And in fact, much of the work on the microbiome, many of the concepts of the microbiome had been developed almost a century ago, but had not been researched and investigated with the modern tools and the modern perspectives that we have now. And so as I was finishing my graduate work, I became interested in why humans harbor hundreds of trillions of bacteria in their intestines, why we have these organisms, and how we tolerate these organisms. And why don't we reject these organisms? Why doesn't our immune system attack these organisms? Because our immune system attacks infectious agents. And that was really the critical question that made me shift my career, staying in microbiology, but instead of working on how the immune system controls infections, I began to investigate, or at least hypothesize, that the immune system itself benefited from these interactions, benefited from interactions with microbes. And so I moved on
What
to...
made you, that's a huge leap. What made you, forgive me for butting in, but I mean, when, like you said, everybody else was thinking in a different direction. Why would you, I mean, what possessed you to go in the opposite direction?
Blind faith, I guess, curiosity, this desire for exploration, but very little data, right? Because again, at the time, 2 decades ago, we knew that these organisms existed, right? I mean, again, as I said, you know, the concept of the microbiome and its potential impact on human health was proposed by Elie Metchnikoff well over 100 years ago, and even by Lou Pasteur and other prominent microbiologists a century ago. But again, in modern times, had just not been studied. And to answer your question, as I started thinking about the trajectory of my career and what I wanted to, you know, to research and what interested me as I moved towards independence, away from, finishing up my training and moving towards my own independent career, I love microbes, I love bacteria, so I wanted to stay in that general area. But again, I have this innate desire to do things that are different than tradition, than the field itself. And I literally read a 1-page article about that reminded me about these bacteria, and it was just very superficial. It just was a more of a commercial article than a scientific one. And literally within minutes of reading that article, I thought to myself, this is where I wanted to take my career. Of course, at the time, because the inspiration, the impetus, if you will, was very spontaneous. I didn't know exactly where I wanted to go, but I just knew I wanted to study beneficial microbes. And then moved on to Harvard, where I first tested the hypothesis that the immune system, its development and its function relied on inputs from the microbiome. We were able to make some discoveries. And then just to answer your question about my career, and then in 2006, moved from Harvard to the California Institute of Technology, Caltech, where I've been ever since. And our lab has studied how bacteria impact the immune system, the nervous system, and the metabolic system in a number of different contexts. And it's important for me to mention, we're basic researchers. I'm a basic scientist. We work in mouse models or animal models of disease. I'm not a clinician. We don't work with humans.
Right. Well, we need you to keep on doing that work so we can use it in our clinical work as well. I mean, your discoveries and those of others have really helped, certainly in my practice and what we're doing to help us to make a difference in the lives of other people. So absolutely.
And that's exactly how we see our role, as to provide potential opportunities for clinicians to test some of these discoveries in human populations of need. Again, we just we develop these technologies, and then we essentially through various different mechanisms release them to the world to be tested in people.
Well, thank you. We have a few more minutes before the break, but we're going to get into your particular area of interest and the company that you've founded, if you want to talk, if you're open to talking about that. But we have about 5 minutes more to talk about what's going on in the fields. I mean, how microbes, let's talk in general. Microbes, we now know, influence our thinking, our emotions, our behavior. You mentioned the metabolism, the immune system. Let's kind of like have an overview of that, because I think that's a very interesting thing for our viewers.
Sure. Connected to what I just mentioned about our research, is most of the research in the field has been done in animal models. And I think, again, that's an important topic, because, not always do discoveries in preclinical models or in animal models translate to people. And I would say, just, in terms of a broad overview of the field, the field is still at a point where, we're testing these hypotheses and testing these potential therapeutics in humans. Where there isn't strong validation that these technologies will be effective in people. right? Again, I know that's a broad statement. There are already some successes, there are already some failures in humans, but the vast majority of what we know about the microbiome comes from animal models, right? And to answer your question, it's become clear that the microbiome, which is composed of on the order of 100 trillion bacteria, bacterial cells that range from 2 to 500 different species per individual, appears to network with our immune system, our endocrine system, our cardiovascular system, our metabolic system, and our nervous system, and probably other organ systems of our bodies as well. And so I would say there's no part of our body that's immune to the potential beneficial effects or the harmful effects, for that matter, of the microbiome. But what we know so far is that, I think there's the greatest amount of evidence that the microbiome, and specifically particular gut bacteria, may have benefits in terms of how the immune system functions. And that might be biologic, meaning that the greatest effect of gut bacteria may be on the immune system relative to other systems, but it's probably, in my opinion, more likely a consequence of that. Those interactions have just been studied more often, and by more researchers, because it makes sense, right? Because if you think about microbes, you start to think about the immune system. It's a natural connection, much more natural than, let's say, the brain, right? And so, we know that particular cell types of the immune system function properly because of inputs, because of guidance from gut bacteria. In fact, there are entire lineages of T cells which only develop by microbial instruction. So this entire, subsets of T cells don't exist when microbes in the mouse, when microbes don't exist. So I think that's
quick one. T cells in the context of COVID-19. Could you give us the context? T cells.
T cells in the context of COVID-19 would be more supportive cells of the immune response to control COVID-19. So the most effective arm of the immune system in controlling viruses are antibodies, which are made by B cells, and T cells support the function of B cells to make antibodies that then control viral infections, including, obviously, SARS-CoV-2.
Fantastic. And microbes affect that development of T cells. Okay, great.
That's right. Microbes affect those development T cells. How that translates to COVID research is still very much ongoing, right? There already have been several reports of the role of the microbiome in either augmenting immune responses that might be beneficial in controlling COVID infections in mice, and certain microbiome profiles that appear to result in individuals that are more protected from COVID-19. But again, all that work is still very preliminary at this point.
I understand.
Pandemic. But suffice it to say, the relationships or the interactions between the microbiome and the immune system are really the most robust, right? Certainly other areas such as metabolic disorders, including obesity and diabetes, have gotten a lot of attention. And I would say that the concept that microbes impact our behavior, or that impact learning and memory, or that impact potentially the function of neuronal cells has been validated, has been shown in mouse models, but I think that at least in human context or extrapolation to human context, that area still remains unvalidated. We still need a significant amount of research. There's a lot of anecdotal evidence. There's a lot of case studies that really strongly suggest that gut bacteria may be involved in, again, mood, affect, behavior, anxiety, depression, even neurodegenerative diseases such as Parkinson's, but we still need more rigorous studies to validate those hypotheses.
Well, studies mean money, Sarkis, and
yes,
that money goes where the interests are. But I mean, I understand you're wanting to be cautious to really make any declarative statements. But if we think about the immune system, I'm speaking to the viewers now, we think about the immune system. The immune system does a huge amount beyond just protecting against sickness and disease. It's a monitor system, it's a surveillance system, it's a Voltron kind of thing. And if anything, we're now beginning to find out, if anything goes wrong with the immune system, it can affect, because it's in touch with all the cells of the body, including the brain and nervous system. The immune system goes out of hand, it could affect. I mean, it's easy to understand that it could affect brain function, it could affect the emotions, it could affect metabolic function. And we know that even from thyroid, when these autoimmune diseases like Hashimoto's disease, for instance, attacks the thyroid gland, you lose your energy, you become, it affects your affect, your thinking, your emotions, and all those things. Same thing with the gut. So everywhere the immune system is, since the immune system is in every part of the body and in every organ and every cell of the body, if anything goes wrong with the immune system, you can easily see that it can also affect those organs and those systems as well. So we're not saying that this is exactly what has happened, because as Sarkis, as Professor Mazmanian has said, you need to have the research to actually verify that. But one can see that simply supporting the immune system and therefore supporting microbes, good microbes, can have a beneficial effect in the long run.
David, I couldn't agree more. And in fact, it's a great way to frame it. And as a physician, you understand that all of these organ systems are constantly communicating with each other. And, we have to study in biomedical research. We use reductionist approaches. We take things apart and isolate, interactions so that we can study them in depth and we can study them reproducibly. But I don't think we do a good job as scientists in putting the pieces back together, if you will, right? And trying to understand how the whole body works, how the entire organism works. But your point is not just well taken. I think that's critical to our thinking moving forward, of not, viewing these interactions in isolation, but in thinking about, the immune system and its, or let's say the microbiome and its ability to impact the immune system may have effects on the entire body and aspects that have nothing to do with direct immunologic function. And so I very, very much agree with you. That means maybe sort of be a quarterback, if you will, from borrowing from American football, of how information and interactions are distributed throughout the body.
Yes, absolutely. Well, thank you, Sarkis, for highlighting that. Quarterbacks. All right, we're going to take a quick break now. We'll be back with Professor Mazmanian in a few minutes, so don't go away. All right, welcome back. I'm here with Professor Sarkis Mazmanian, and we're talking about the effects and the role microbes have in human life and human health. And I think I'm going to have to pause this. All right, welcome back. I'm here with Professor Sarkis Mazmanian, and we are talking about the effects of microbes upon health, human health as in general. And as I mentioned in the introduction, we were talking about how we at the Brain and Body Foundation, we started really with autism, children with autism, under the supervision of the federal government of Nigeria. We were treating brain disorders starting with autism. And we began to see the role bugs had to play in them. And so we started looking for probiotics. Probiotics are beneficial bacteria, which have been shown to help in several different areas, as we discussed earlier. And we've got some good ones, and we tried them, and they worked well. And we got some, I would say, beneficial benefits. And when we increased the dose, we got even greater benefits. But there was always something missing. And we started looking, and we found out that there were specific microbes that are prominently absent in cases like autism and children on the autism spectrum. And we found out from our guest today, I actually attended one of his lectures, that those microbes, when put back somehow into the body, a lot of those autistic symptoms and behaviors are, shall we say, removed?
Yeah, or improved, yeah.
Or improved. Yeah. A lot of them, just from one microbe. So I was like, where can we get this microbe? And that's how we found a way to reach out to Prof, and one thing led to the other. And so here we are. So let's talk about Bacteroides fragilis.
Thank you, David. So as we were doing our research over the years, and thinking about all these different interactions, but in particular, the gut-brain connection, we wondered whether or not we can essentially access this rich communication between the intestines and the brains through microbes. And so, maybe the first step is I think a lot of this, what I'm about to say, is intuitive, but at least I wasn't thinking about it on a regular basis, but just how much our brain and our intestines actually communicate with each other. So if we think about our senses, right, sight, smell, hearing, taste, and touch, our visceral senses, I mean, these are all inputs into the brain. And the brain uses those inputs not just to sense the environment, but to make decisions. But I would argue the largest molecular input to the brain is through the gut, right? So in terms of actual molecules that enter our body, our digestive system evolved just for this reason, right? Is obviously to bring in nutrients to nourish our bodies, but also brings in a rich chemical library of molecules that circulate throughout our entire body and into our brain. And so again, many molecules that are found throughout our bodies, including our brain, are products from bacteria, generally dietary products that that were metabolized or produced by bacteria are what we absorb. In fact, I've read studies that say somewhere along the lines of 90% of the molecules that we ingest, that entered into our body were first metabolized by bacteria, right? And so essentially bacteria alter what we eat, and then we eat their byproducts. And so this really was fascinating to me to think that there are so many, that there's such a huge impact of the intestines on outcomes that are regulated by the brain, whether that's behaviors, whether that's neurodegeneration, whether that's decision making, memory, and cognition. And so that was the initial insight, right? It was to think about using bacteria to essentially access this highway between the gut and the brain. And so again, as I mentioned, our work is in mouse models, and we initially used mouse models of autism to test these hypotheses. And in theory, it could have been other disease indications, models, let's say schizophrenia or depression, where we could have started. The reason why we started with autism was very logistic, is we had a very good collaborator at Caltech who had developed mouse models of autism, and so we had access to them. And so there's a logistical component to it, but of course there's a scientific and health component to it. Is that about half of the autistic population have some form of GI issues. So many children with autism, which is a behavioral disorder, have constipation or diarrhea or abdominal cramps and bloating that have been reported. Again, a lot of this is anecdotal. There are several clinical studies now that validate this. So again, there was a medical rationale for thinking of using autism to test our hypotheses. And so we, through a series of studies, showed that in this mouse model of autism, there was a depletion of particular microbes. And this was, obviously interesting to us, because what it does, it creates an opportunity to test a concept that maybe the absence of bacteria is a risk factor for disease. So again, this connects to what we had just discussed a few minutes ago, right? Is that, bacteria aren't always bad. So if you have good bacteria, and if you're missing a good bacteria, then the absence of that good bacteria is your risk for developing a disease. Yeah. And so then that creates the opportunity that I just mentioned of now supplementing, and as David alluded to as well, of restoring what would be, let's say, a healthy microbiome by administering particular bacteria. And so we use Bacterius fragilis for various different reasons, is that it had both immunologic properties and it had GI restorative properties that were very attractive to us. And so as I mentioned, in autism, which was also reproduced in the mouse model, that there were GI symptoms. So these mice had features of constipation, and they also had barrier defects, which some may call leaky gut, but essentially the intestine was absorbing things that it wasn't supposed to absorb. So even toxic molecules, which in a healthy gut are excluded, were being found in the circulation of the animals. So our hypothesis was that if we potentially repaired the gastrointestinal systems, if we made the intestines work better, would we then see benefits in terms of behavior? In fact, we did. So we treated the animals with Bacterius fragilis, which again, our prior work had shown had the ability to improve gastrointestinal function And not only did it improve gastrointestinal function in the animals, but it improved almost all of their behavioral abnormalities or their behavioral deviations as well. And so autism itself is diagnosed by deficits in social communication, which includes both verbal and physical communication, as well as repetitive behavior. These features can be modeled in mice. So mice don't get autism. Autism is a human quality, but mice socialize, so they socialize differently if they have features of autism, or they communicate differently than a control animal. And so what we were able to show in this mouse model is that Bacterius fragilis improved communication, it improved anxiety, it improved some of the sensory issues, it improved the repetitive behaviors that these mice were exhibiting, which again, I think speaks to the fact that it has this broad activity, not that it's, sending one, and what I'm about to say is my opinion, it's we have no data one way or another, but that it's not as if the organism was, sending specific chemical signals to one neuron in the brain that controlled repetitive behavior, another neuron that controlled vocalization, but it had these more broad, physiological effects on the animal, which again, we trace back to its improvements in GI function. And so that's how we initially validated and ultimately published in 2013 that Bacterius fragilis, at least in a mouse model, had very broad effects in improving altered behaviors.
That is amazing. That is one single bacterium. I mean, that's bizarre. And if I understand this correctly, you were at first ridiculed for coming out with that. There was a lot of kickback.
Well, there's always skepticism, right? When there are advances that's aren't traditional, that aren't linear in terms of thinking, right? And so what I mean by linear is our research wasn't the 50th step in a research project. It was the first step in this discovery, and so whenever there's sort of this disruptive technology, I think it's very healthy to be skeptical, right? Because there are a lot of discoveries, there are a lot of claims that ultimately aren't validated, and so if we just pursued every potential exciting discovery, then I think we'd be on the wrong track a lot. And so the concept that one can treat brain disorders via the gut, via bacteria, 10 years ago or 8 or 9 years ago, the fact that it was met with skepticism, I think is natural. I think that's the scientific system to not allow every claim to just be believed to be fact. But ultimately what happens is that if in fact that disruptive technology, that discovery has scientific merit, then it will be validated by others. It will be reproduced by other groups, or by other organizations, and then ultimately developed to where we have both more understanding of how that system works. So if you described it, if I do A, then B happens. Well, the question was, well, then how does that happen, right? So if you start filling in the gaps, right, that builds more evidence, more credibility for that discovery, because now there's substance in understanding how that happens. But ultimately, we want to test these concepts in people, because I think to me that is the best way to validate initial biomedical research, is to show that in a very different context in humans, that you're able to essentially reproduce the findings from the preclinical mouse studies. And so now I think we're well on our way to getting an answer. We don't have that answer yet, but we're well on our way to getting the answer of whether this organism and the mechanisms and biological mechanisms used by this organism may be effective for treating autism, based on human trials. And so, as David has mentioned to your audience and has interviewed Stu Campbell from Axial, I co-founded a company to do just that, was to take this technology from our academic laboratory, where we don't do human research, and to now develop it and test it in the context of human trials. And Axial has completed an initial trial in autism where with technologies that I'm discussing, where the results were quite positive and quite encouraging. And even then, the first human trial doesn't mean that you've made the ultimate discovery. It doesn't mean you've solved the problem. You've now embarked on a new road, a new journey, right? But now entirely in humans to ultimately reach what would be our objective or our goal of getting an approved therapeutic for behavioral disorders such as autism.
And I can't wait for that, Sarkis. I honestly can't wait. I'm like, when can we get this thing? I mean, what's the delay, right? And of course, like I said, we have lots of people who would love to be a part of the study. So if we can make it happen, we're all in. We're all in. So we're watching very, very closely and how this thing goes. We have a few minutes, at least. I'm sorry, we have about 2 minutes left before the end of the show. But I definitely want to hear. Do you have any final you want to leave our audience with?
Well, at the end of the day, there are many ways to treat symptoms or treat disorders once they've manifested. And many of, we do that in biomedical research, we do this, but there are many disorders, many conditions. It doesn't even have to be a disease, right? So if you're lethargic or, you're having a concentration or memory issues that it's not a disease, it's not a disorder, right? But it affects your quality of life, affects your daily activities. Those conditions are being addressed, but those are more, these are very challenging, but maybe the final thought would be is that as we understand more from these types of research questions, one of the themes that always keeps coming back to us is that ultimately the effects of microbes are dependent on the nutritional and biological state of microbes. So in other words, if a microbe is going to have a therapeutic effect on a human as a concept, that microbe itself should be healthy. And how are microbes healthy is if they are getting the nutrients that they need for their proper physiology. All this is a long way, David, of saying that a healthy diet and a healthy lifestyle ultimately, has huge impacts on health, and those impacts may be largely through the microbiome as well. And so until we get to the point where we understand enough about biology, enough about human physiology to be able to, give a treatment in a bottle, I think we can make a lot of gains in the interim just by living very, very healthy lifestyles. And so what I just said is obvious and is intuitive, but sometimes I need to remind myself as well that I can get a lot of benefits, not from prescription drugs, but just from eating healthy foods.
Absolutely. I totally agree with you. But then there are also others who cannot recover from their illness and disease without some kind of intervention or help. So the healthy lifestyles are extremely important now. We really impress upon people. But also, we also need help from time to time, which is why we're so happy grateful for what you're doing and people like you who are doing this kind of work. And anything we can do to help push it forward, please let us know.
Thank you, David, and congratulations to you and your organization and your foundation for all that you're doing. This is not what you've set out to do is not an easy task, and I can already appreciate that from a distance. But a very, very worthwhile one. So thank you.
I appreciate it, Sarkis. God bless. And thank you again. Well, thank you, folks, for joining us. Remember, these videos are available on our site and on the NTA website as well. So stay tuned, stay safe, and keep on living a healthy lifestyle. God bless. See you next week.